In this video, we are going to be learning how to read molecules. So when you're reading molecules, you want to first look at the skeletal structure, and then you can determine what type of molecule it is and how it behaves. So first, what you want to do is you want to count the heteroatoms. So when you are going to look for atoms that are not C or H, and these atoms will be usually the elements of O, oxygen, nitrogen, phosphorus, or sulfur. And these are the atoms that define the behavior. Now, your second step is to look for key patterns. Like, for example, OH is a pattern, C double bonded to O, phosphate, or a ring. And now, after this, you're going to ask the key question. Is this molecule polar or non-polar? And from this, you can determine if it is a sugar, if it's amino acid, or a peptide. If it's a if it is a nucleotide, if it's a lipid, if it's a cofactor / regulator, but just from its polarity. So first, what we're going to talk about is the hydroxyl group. So this is the hydroxyl group. So usually what this means is, if you see this, it's usually going to be like a sugar. And let's make this down a little bit. So hydroxyl is our first pattern. This is going to be one of our functional groups, hydroxyl. So this, for example, you can think of ROH, and you can think of glucose. That's C6H12O6. That's going to be HOOH. OH, O, and H. So it's going to be carbon double bonded to an OH. And just think about, like, you're just trying to identify these, like, dangling OHs. And they're usually on sugars or alcohols. So some of the properties are that they're polar, they can donate and accept H bonds, and they increase water solubility. So if you see many OH groups, it's going to be sugar-like. And if you see many OH bonds, OH groups on a carbon chain, so for example, on a carbon chain or a ring, then what you want to do is think it's a carbohydrate. Okay, now let's move on to carbonyl. So this right here. This is going to be a carbonyl. Okay, so when you have carbonyl, it's going to be like O, C, and then you have two things right there. So this is a carbon double bonded to an oxygen, and some things that we have, like, that's like with this is that this matters because it's going to be like strongly polar. It's also going to be very reactive, and it's also central to metabolism. But carbonyls, they can split into two subtypes. Now, after this, we have our aldehyde. So aldehyde is going to be a carbonyl at the end of the chain. It's CO bonded to H. And what you have to identify are these aldose sugars, and that's going to be like glucose. So remember, if it's at the end of the chain, it's an aldehyde sugar. So you can think of, you have like COH, or you have RCOH, where R equals H, alkyl, or aryl. Now, after this, we can move on to ketone. Ketone. This is going to be a carbonyl in the middle of a chain. So this is basically C double bonded to O, bonded to 2 carbons. So remember, if you have a middle carbonyl, that means it's a keto sugar. And this was an aldehyde sugar. Aldehyde sugar. Now, after this, let's examine carboxyl groups. So that's going to be COOH or COO. So, carboxyl, that's like this. So you have and for C, you have CO and you have OH. So that's going to be, we can represent this blue. There, and so basically that's going to be a CO and OH on the same carbon. So some characteristics of this are that they're acidic. They are also often deprotonated in cells. So that's COO minus. There's also strong H bonding. So this means amino acids, also means fatty acids, and also metabolic intermediate. So think amino acid or an acid. Now let's move on to the amino group. So that's NH2 or NH3. So you can think of the amino group that is, you have N, H, or you have, you can just, I'll just write this here. Or actually, no, that's fine. I need to write that out. We have NH3, so that's usually they look something like this, and you'll usually have like gray for, okay, that's not gray enough. And you have green. So now, and also for, this is your amino group, and then if you have amine, that's like R, N, H, H, and then it's like the same thing. You just have R off to the side, and you have your color that's going to be a little darker. So this is usually a nitrogen bonded to carbon, and it is often protonated in your cells. So it's usually basic. You also can accept protons. You have hydrogen bonding. So remember that amino plus a carboxyl, that's going to be equal to your amino acid backbone. Now, amide. Amide is CONH. This is going to be a carbonyl directly bonded to nitrogen. Now, this is usually found in peptide bonds, in proteins, and in stable linkage. Now, let's talk about ester. Okay, for ester, this is going to be a carbonyl plus the oxygen linking two carbons. Now, an ester is usually going to be like this. You have your carbon and you have your other, like that. Now, it's usually found in lipids, triglycerides, and in phospholipids. Remember, if you have a long hydrocarbon chain plus ester, that's going to equal to a lipid. Now, let's talk about ether. So, you can think of this is going to be an oxygen between two carbons. And you also have no carbonyl nearby. So this means that it's stable, and it's also going to be common in sugars. And also these sugars, they are usually going to be connected by either like glycosidic bonds. Now, after this, we can draw some places where we can identify some ethers. So, like, let's think about if you have HCOH, you have CCO, C up there, OH, you have OH. Okay, hold on. You have OH attached here, you have a H here, you have a H here, you have a OH here, you have a CH2OH there, right? So this is going to be your COC right here, and an H up here. That's your ether group right there. Okay, now after this, let's move on to the phosphate group. Phosphates group, okay, and this is going to be mono, di, and also tri. So what is this? This is a central P with 4 oxygens. So it's often negatively charged. and it's highly polar. You have energy storage. You have strong electrostatic interactions. And you remember that phosphate is equal to energy, signaling, and nucleotides. So, for example, you can think about there's a whole bunch of phosphates in this. So we have one here, one here, one here. You have a here. That's gonna be negative. You have this. Then this here that's attached to a base. You have 2 hydroxyls here. And basically this is going to be your nucleoside. This, at this level, this is going to be a nucleoside monophosphate NMP. And then here it's going to be a nucleoside diphosphate. That's an NDP. And then this is going to be a nucleoside triphosphate. So here we can see all the levels: phosphate and NTP. These are all different types of nucleotides. Okay, you should also be familiar with the ATP-ADP cycle. So that's going to be, you have this A, so this is going to be your ribose. Up here you have your triphosphate. And down here, basically what happens is, this is going to be a cycle, so then you have your phosphate that breaks off, and then when you break off that phosphate, it releases energy for the cell. And then after this, what you're left over with is, it's the same thing, but just one phosphate gone. So that's going to be your diphosphate. That's going to be your ribose here, and here is your adenine. So this is going to be your ADP. And to start the cycle again, that's how you add back your phosphate. And then you have energy absorb for food, from food. So now after this you have your sulfhydryl and your disulfide. and your disulfide bonds. So that's going to be like, or like, for example, you have you can see that you have your SH, and what this means is your sulfidal means that it's reactive, and also it's found inside cysteine. Okay, and then your disulfide bonds, that's going to be when you have two sulfurs bonded. What this does is it gives you protein stability. Now for aromatic rings. So this is going to be a hexagonal ring with alternating double bonds. It's hydrophobic, and it also is stacking interactions. And overall, just think about its binding structure and also signaling. So you can think about, for example, okay, highlight that, that's like benzene. Also, benzene can be this too. Just double here, double here, double here, double one up here, here, and here. Okay, so this is an important one. It's hemiacetal versus acetal. This is very important for sugars. So hemiacetal is going to be OH and OR on the same carbon. And you also have a free sugar N. Now for acetal, you have two OR groups, and you have a locked glycosidic bond. And when you have a free hemiacetal, that means you have a reducing sugar. So again, let's review this. If you have, okay, back up here with hydroxyl. Hydroxyl means you have many OH. That means you probably have a sugar. Now, if you have an okay, let's actually just like write this here then. Okay, sugar means many OH. Amino acid means amino plus carboxyl. If you have a protein, that's probably going to be repeating amides. If you have a lipid, it means you have long C chains, and you also have ester. If you have a nucleotide, that means you have a phosphate plus a sugar plus a base. And if you have phosphate alone, that means it's probably for just energy or signaling.